Hello everyone, welcome back to my channel. Today we are going to discuss histology of lung. So first we'll make an outline. So we are going to draw one representative bronchi, the bigger conducting system, the smaller one the bronchiole. Then we'll just share the background where we are going to write the rest of the structures, alveoli, alveolar ducts and all. So this we have to make an outline. So when you're drawing, make sure bronchi is a part of the lung. So don't draw half of the circle as bronchi. It should be very small and it should be a one part of the whole lung. Alveoli should be more. As we know, trachea histology already been discussed. So we have trachea divides into two bronchi. They are called primary bronchi for each lung, and each primary bronchi will divides into the right bronchi divides into three for the three lobes. That's called lobar bronchi. The left side that divides into two. Okay, each lobar bronchi again divides into segmental bronchi, 10 on each side for each bronchopulmonary segments. So as soon as the bronchi, the bigger one, that enters the lung, the layers will keep changing. As we know in the trachea, almost similar to trachea, but there is a minor change. We have the appearance of the smooth muscle here, and the cartilage which was C-shaped in the trachea becomes broken and irregular as it comes to the bronchi. So basically we have 5 layers in the bronchus. The first layer is almost similar to the trachea epithelium, that is pseudostratified ciliated columnar epithelium with the goblet cells and all, and the lamina propria. Together they call the mucosa. So then we have a smooth muscle layer which will be very prominent. Then we have a gland layers where we have a serous and mucous glands. It is usually in between the cartilage and the smooth muscle layer. And it is within the submucosa, the connective tissue layer with the glands. That's the third layer. The fourth layer will be the cartilage. Initially, which in the trachea, which is we have in the C-shaped cartilage, will be broken down, and it is present throughout the circle, the circumference of the bronchi. So this keeps the bronchi patent, the open. So we have to write similar way: hyaline cartilage, the hyaline matrix, transparent matrix with the cell nest, the chondrocyte present in the cell nest, and the matrix which is hyaline, the bluish color, the territorial and interterritorial matrix. The cartilage will be in small pieces. If the cartilage is present, that's the key structure, that is a bronchi. Bronchi are the bigger branches. It is almost similar to the tree branching. So wherever the branches of a tree like that, the brown color, the stem, that's a bronchi. So the last layer is the adventitia, which connects the bronchi to the underlying structure, either to the alveoli or the underlying lung parenchyma. So then coming to the bronchiole, as the bronchi keep on dividing, when the diameter becomes less than 1 millimeter, they are called bronchiole. So the lumen also will decrease. As we know, the pseudostratified ciliated columnar epithelium, the epithelium slowly keep decreasing. The goblet cell disappears. The columnar cells become low columnar, and then the columnar will become cuboidal. So in the bronchiole, we have the bigger bronchiole having the tall columnar with some having the goblet cells, whereas the smaller bronchiole, that is a respiratory and terminal bronchiole, the epithelium will be low columnar or cuboidal epithelium. So next to that, you have a very small lamina propria. Then we have a smooth muscle surrounding that. Smooth muscle is very important here. This smooth muscle will regulate the amount of air going to that set of alveoli. So this constrict and relax and regulate the amount of air going through this bronchiole. So only this layer, we don't have any cartilage, no glands, and even the goblet cells will be absent. That's the main difference in the bronchi and bronchiole. So in the bronchiole, we don't have any glands, no cartilage, no glands, no goblet cells, and smooth muscles are prominent. Next to that, we have adventitia. So, and the diameter is also less than 1 millimeter. Till the terminal bronchiole, they are the conducting part. Then start the respiratory bronchiole. Respiratory bronchiole is like a branches of the tree where your leaves are directly attached. Leaves are like alveoli. So respiratory is also tubular structure, respiratory bronchiole, but in between some alveoli directly open into them. So you have a gap there. So and the lining epithelium will be cuboidal epithelium. So wherever the gap is there, the alveoli are directly opening into them. So that is the representative respiratory bronchiole. Respiratory bronchiole will lead to alveolar ducts. Alveolar ducts are tubular structure, but in the wall it doesn't have any walls. Instead, they have alveoli as their walls. Then the alveolar ducts will lead to alveolar sac. Sac is a space surrounded by alveoli. Then is the alveoli. Alveoli are the dilated polyhedral structure. I'm just representing a few blood vessels, the bigger one. Even the smaller capillaries will be there surrounding the alveoli. The other side, we are representing the pleura, the pulmonary pleura which is covering the lung, visceral pleura. Then most of the places, the remaining places, we have to fill them with the alveoli. The alveoli are the polyhedral structures. Don't write angled one. They are like a bunches of the grapes. If when they're clubbed together, you can see the different angles, shapes, but there is no angles in them. The surface can be polyhedral. So don't leave much gap in between them. Even the minor gap is fine because surrounding the alveoli, we have a network of capillaries. So where the blood will be running, and the blood in the capillaries and the air within the alveoli, there is an exchange of gases will takes place. So even the minor places, the angles and all, you can shade it with the pink pencil like that only. That represent the small spaces surrounding the alveoli, which contains the capillaries, and some amount of connective tissue also will be there. So we have some certain type of cells in the bronchiole called Clara cells. They are nothing but a cuboidal cells, similar which functions in the type 2 pneumocytes. They are the one which secretes the surfactant-like structure and prevent the collapse of the epithelium in the bronchioles. They are the Clara cells. Then come to the alveoli, we have two types of epithelium basically. Type 1 pneumocytes, even though the number is less, they have a very broad surface area, and they are the squamous epithelium, and they are the responsible for the air exchange barrier, blood air barrier, the exchange of gases between the air and the blood. They are the squamous epithelium. Another type of epithelium is type 2 pneumocytes. They are the cuboidal epithelium. Even though the number is more, the surface area is less. That's why you will see very rarely the epithelium. They will be cuboidal. We'll write some representative cuboidal cells in the alveoli. They are the one which secretes the surfactant. So basically lung tissue, we have to represent one bronchi which is just a part of it. Don't cover whole bronchi in the complete circle, just a part of it. Then you have to represent bronchioles, then respiratory bronchiole, alveolar duct and a sac, then some blood vessels here and there. In the alveoli, don't make it angled one. Draw polyhedral. Sharpen your pencil and keep a flat nuclei representing the type 1 pneumocytes. And here and there you can keep one round nuclei representing the type 2 pneumocytes which secretes the surfactant. So lung is easy to identify in the slide. We'll see plenty of alveoli, the air-filled spaces. Basically you have to differentiate between bronchi and bronchiole. If there is a cartilage, that's bronchi. If no cartilage and you can see the clear smooth muscles surrounding the small lumen, that is bronchiole. These are the type 2 pneumocytes which I am drawing. For easy understanding, I have added a pic of labeled structure. If you like the video, give a thumbs up, share with your friends, and subscribe to my channel. Thank you.